propagation. So vegetative propagation, they classify into two types: natural vegetative propagation and artificial vegetative propagation. So this natural vegetative propagation, it takes place by root, leaf as well as stem. Example for root, that is sweet potato; leaf, bryophyllum; rhizome, example turmeric; tuber, example potato; and bulbs, onion. So they produce small bud, and these bud develop into new organism. So the importance of natural vegetative propagation, that is plants produce asexually through their vegetative parts. So the new plants form are genetically identical to their parents. The second part, that is artificial method of vegetative propagation. So artificial method classify into three types: cutting, grafting and tissue culture. So in cutting, the part of any vegetative part of plant having one or more buds is used for propagation is called as cutting. They further classify into stem cutting, example rose, sugarcane; leaf cutting, example bryophyllum; and root cutting, example apple, blackberry or sweet potato. So these parts produce small bud and finally they develop into new plants. The second, that is, these are grafting method. So in grafting method, there are two type of plants. The upper part of plant is called as cycon, the lower one called as stalk. These are two different plants. So part of these two different plants, which are genetically mostly advanced or they are with good character. So these two parts, they can be joined with the help of thread or tape. So these bud grafting, which is also called as budding. In budding, the bud grafting is in which only one bud is joined on stalk. This is T-shaped cutting. The lower part is called stalk, upper part is called as cycon. There is T-shaped budding. So this bud, there is only bud from another plant. It is given and they are attached with the help of thread. This is T-shaped budding. In T-shaped budding, only single plant is there and the bud from other plant, they can be mostly add into this T-shaped cutting. The next one, that is tissue culture. So in tissue culture method, the part of the plant, they are cultured in nutritive medium in aseptic condition to produce complete plant or plant part. So importance of tissue culture, it is a method by which small amount of plant tissue is carefully grown to give many plant. So this is mother plant. This mother plant, they cut into small pieces. This small piece is called explant. Explant, it is culture in tissue culture. Explant divide and redivide to produce callus. After callus, they grow into shoot and root. This process is called as organogenesis. And finally, they produce multiple plants. So these are the method of artificial vegetative propagation, that is cutting, grafting and